Hello, we're continuing with my notes for cell biology in a nutshell and life science. We're learning about the reactions that go on in an organelle called the peroxisome, which is found in eukaryotes. And peroxisomes specialize in breaking down large molecules, especially fatty acid um, molecules, long chains, and converting them to the fatty acids, which are found in such things as the lipids of the seeds, for example, and converting them to sugars. The sugars, which are then used for growth and development. So fatty acids produce acetyl-CoA molecules, which are turned into succinic acid by the peroxisomes and then converted into glucose, which is a sugar. The glyoxylate cycle, which is what I just described, the conversion of fatty acids to glucose, does not occur in animal cells, only in plants. So, therefore, animal cells are not able to convert fatty acids into carbohydrates. Only plants are. So in leaves, for example, this occurs. So for example, in the germination of seeds, this occurs where the lipids in the seeds are converted into sugars so the young plant can sprout. Also photorespiration, which is the opposite reaction of photosynthesis, producing carbon dioxide instead of oxygen. It takes the oxygen and releases CO2, which is the opposite of photosynthesis, which takes CO2 and releases oxygen. But this is done because in carbohydrates, so this reaction where oxygen is used is oxidation. And oxidation is important in, for example, breaking down such things as methanol and alcohol, which is oxidized. So peroxisomes oxidize methanol as well as fat, and they break down fatty acids as well by oxidation as well, yielding acetyl-CoA. So oxidation breaks down fatty acids, the alkyl chains, and shortens them 2 carbons at a time, converting the fatty acids to acetyl-CoA, which the acetyl-CoA is then exported to the cytosol and used for various biosynthetic reactions, such as making of sugars, etc. So in plant cells and yeast cells, this occurs in peroxisomes. In mammalian cells, this kind of oxidation also occurs in the mitochondria as well as the peroxisomes. So a variety of substrates can be oxidized, such as phenols, formaldehyde, formic acid, alcohol, etc. So usually H2O2, which is peroxide, hence the name peroxisome, is used in oxidations. This H2O2 is generated from various enzymes in the peroxisome, and it helps to oxidize the various substrates. The reaction is H2O2 plus the substrate H2 yields the substrate alone, and two waters are released. Peroxisomes in this way help detoxify various alcohol and other toxic molecules, especially in liver and kidney cells. Ethanol, for example, is oxidized to acetaldehyde. So when H2O2 accumulates in cells, this converts this catalyzes this causes the reaction which we just mentioned. So the H2O2 is converted to water by adding the substrate needs to be broken down. So substrates that have alkyl groups that can be broken down by this reaction via the peroxidative reaction. Alkyl groups include methyl group CH3, ethyl group CH3, CH2, propyl groups CH3, CH2, CH2, isopropyl group CH3, CH3, butyl group CH3, CH2s, three of them, S-butyl group CH3, CH, CH2, CH3, isobutyl group CH3, CH, CH3, CH2, T-butyl group CH3C, CH3s on three sides. So an alkyl group is defined as a functional group made up of carbon and hydrogen entirely, where the carbon atoms are chained together by single bonds. The general molecular formula for a simple alkyl group is C to the N, H2 to the N plus 1, where N is the number of carbon atoms in the group. If the chain is branched, the alkyl group gets the name according to the chain. So the length of the alkyl chains determines the properties of organic materials and enhances their performance in various ways. Therefore, the steric effects of the molecules benefits the various reactions that they are part of. So these are examples of the alkyl groups. Alkyls. A simple alkyl group is a functional group made up entirely of carbon and hydrogen, where the carbon atoms are chained together by single bonds. Its general formula is we went over C to the N, H2 to the N plus 1, where N is the number of carbon atoms. If the chain is branched, the alkyl chain gets the name according to the chain. The length of alkyl chains determines the properties. An alkyl substituent is an alkane missing one hydrogen. The term alkyl is intentionally unspecific to include many different possibilities. We'll continue in the next part. Thank you.